Professor Jim Vallis and I have the delightful responsibility of introducing today's speaker. Today's speaker is Professor Matthew Fisher. He's from the University of California, Santa Barbara Physics Department. And he's also, oh, okay, we have some surfers here. And he's a scientist at the Remarkably Institute for Theoretical Physicists, where you've been for quite a while, I think, at this time. Matthew, for just the lineage, Matthew came from the University of Illinois, your Urbana-Champaign, where he got his PhD. Yes, yeah, there are people from Chicago too here. And he worked at IBM, T.J. Watson facility in Yorktown Heights, New York, for probably almost as many as 10 years, but not quite that long. And then went on to Santa Barbara from there. Professor Matthew Fisher is very well known for being a broad thinker. He has focused a lot on correlated electron systems and worked on things that include high temperature superconductivity, spin liquids, fractional quantum Hall effect, and something very near, dear, and close to my heart, the superconductor to insulator transition. His approaches to all these problems are ones that have a broad appeal. And when I say that, a broad theoretical appeal, so that people in high energy theory and cosmology pay attention to what Matthew's been doing. Experimentalists really love Matthew's stuff. We see it as being creative, imaginative, thought-provoking, rigorous, and enabling. You can create community in physics by doing science in a way that invites others to do things, and if you give others tools to analyze things, like scaling tools, as many people have done, you actually pull more people into the discussion. So it's truly a great pleasure to introduce you to Matthew, who is also a fellow of the National... I don't know.
Felony, not yet. Not yet.
Yeah. Oh, sorry. A fellow of the National Academy of Sciences, a fellow of the American Academy of Arts and Sciences, and a Buckley Prize winner. Thank you.
Thank you, Jim. That's so exaggerated, I couldn't even recognize myself. But it's certainly a pleasure to be here. I did want to mention that I was an undergraduate at Cornell. So I see, Ithaca. But yeah, so it's a real pleasure to be here and to have the opportunity to tell you a little bit about some things I've been thinking about for the last 5 or 6 years, which are really, I would say, off the beaten path. This is kind of pretty far outside of the mainstream, but I want to try to convince you that maybe it doesn't need to be that far outside of the mainstream. And so, in particular, the title then is Quantum Processing in the Brain. And notice the question mark, and that's important. That's what it's a question. We want to answer questions, and this is the question I'm trying to focus on eventually answering. And another title could be, Are we quantum computers? So think about in your own head, are you a quantum computer? So let me tell you a little bit the story behind how I got into this question, are we quantum computers? And the story doesn't start with quantum mechanics, but it starts with lithium. And lithium is remarkable. Lithium's an atom. It's the third simplest atom. Lithium comes in 2 stable isotopes. Lithium-7, which has 3 protons and 4 neutrons, which is a relative abundance of 92%. So naturally occurring lithium is almost all lithium-7. Lithium-6 has 3 protons and 3 neutrons, but that's the rare isotope of lithium at 8% abundance. But in addition to being a very simple atom, lithium is a remarkable drug. It's a drug for bipolar disorder, and it's been used in the United States since the mid-1970s when it was FDA approved. And as I say, it's one of the most effective drugs for bipolar disorder. It's also used to augment antidepressants for people that suffer from depression. So I've been very interested in psychiatric pharmaceuticals, and I've tried a number of different drugs over the years, including lithium. And I should say that, if you need it, lithium's a good atom. You should try it. Nothing to be ashamed of there. Sometimes I ask, who here knows someone that's taken lithium? Okay, what I won't ask is who here takes lithium, although I take a little bit of lithium, so, but there's still a taboo in this country about psychiatric illnesses, and that's one thing. It's kind of a shame. And any case, I've been interested in psychiatric illnesses. I've been interested in the drugs, which are very effective, in particular lithium. And so, it was in 2013 I sat down to ask, how does lithium work? And in fact, how do any psychiatric medications work? And the bottom line is we don't really know. But when I started learning everything I could about lithium, I had what seemed like kind of a crazy idea, which was to feed lithium isotopes to rats. So even though lithium naturally occurring is almost all lithium-7, it's possible to isotope purify lithium and buy lithium-6 from an isotope company at 95% purity. And so the idea for the experiment was to take some rats, feed one set of rats pharmacy lithium, which is essentially lithium-7, another set of rats lithium-6, a control set of rats not fed any lithium at all, and ask whether there are any behavioral differences. Okay, and in the most remarkable day of my scientific career, I found that this experiment was actually tried in 1986 with results so surprising that we should all be suspicious of their conclusions, but I will tell you what their conclusions were. So basically, they did this very experiment where they took, I think it was 24 rats. They divided them into 4 groups of 6. These were female rats. They put them on lithium, pharmacy lithium, lithium-7, the rare isotope of lithium-6, the control rats for 10 days. They impregnated these female rats, and during the 20-day gestation period, continued to give them lithium, the isotopes of lithium. Then they gave birth to the pups, and most of the experiment was looking at the development of the pups. But what caught my attention was the one table which described the mothering behavior of the rats. And they looked at various mothering behaviors like nest building, nursing, grooming of pups, retrieval of pups, grooming of self, state of alertness, cannibalization of pups. I don't know, that's not really mothering behavior, but so there's 3 columns here. There's a 4th one I'll show you in a second. The first is the control rats, and this is very suspicious. All of the control rats were average. Was this experiment done blind or double blind? Or what does it even mean to be double blind when you're doing experiments on animals? I'm not sure. But in any case, this is very, it is somewhat suspicious. But let's just look what they reported. So for pharmacy lithium, which is 92% lithium-7, or lithium-7, which here is 99% lithium-7, what they found was nest building was absent, nursing was infrequent, grooming of pups was infrequent, grooming of self was absent, reaching for food was infrequent, state of alertness was low. So generally, according to this data, their affect was flattened, and and that's maybe consistent with what you would expect from the human experience in taking lithium to counteract mania, where it sort of flattens the affect. But then the really remarkable column was this lithium-6, which is this rare isotope of lithium. And the nest building was excessive. Nursing was very frequent. Grooming of pups was excessive. So these were the cleanest pups in the world. Retrieval of pups was excessive. I'm not sure how that can be excessive in any case. And then what caught my attention, state of alertness is very high. That's the drug I want. I don't dare try it. But I did a couple of things when I saw this data. I said, well, I'm not sure I believe this, but why don't I patent it anyway? So I patented lithium-6 as a possible psychiatric pharmaceutical. I've tried to reproduce versions of this experiment, which I could tell you about at the end if I have time. And I also said, well, let me just assume it's correct and try to understand how it's possible that the different isotopes of lithium have a differential behavioral manifestation in rats. Because basically, there's a small mass difference, but really what one is saying here is that one less neutron in lithium-6 seems to make all the difference. Now, a neutron is a tiny particle. It's quantum mechanical. And so this got my juices flowing, and I started thinking, good Lord, could it be that there is some quantum processing going on in the brain? Okay, could it be that we are, in fact, quantum computers? Now, this is a kind of a difficult field to enter. First of all, the field doesn't really exist. But to the extent that it does, it's got a really bad precedent. So these are 2 books from 1989 Maybe you've read one or the other. Quantum Healing by Deepak Chopra. So Richard Dawkins says Chopra uses quantum jargon as plausible sounding hocus pocus. Well, that's not very good praise, is it? So Roger Penrose wrote this, who's a mathematical physicist who collaborated with Stephen Hawking. You've probably heard of him, who wrote this, quite remarkable book, The Emperor's New Mind. And he argued that classical physics is inadequate to explain consciousness, and that somehow the brain must be harnessing quantum mechanics, which is operational, he argued, in microtubules. Now, Patricia Churchland, who's a neurophilosopher, panned Roger Penrose's books. And her quote is, "Pixie dust in the synapse is about as explanatorily powerful as quantum coherence in the microtubules." So that's pretty damning as well. And now I do wonder how much quantum mechanics Patricia Churchland knows. She's not a quantum physicist, but anyway, it's easy to criticize other people's work. But I will say that when this book came out, I was just out of graduate school, and I read it, and I was, pretty intrigued by it, but I didn't really know what to make of it. But there's a good reason to be skeptical about the conclusions here in Roger Penrose's book, and the reason is the following: biological processes are slow. So, for example, if you ask what's the rate of a protein to fold, it's a characteristic biological process, the folding rate is about 10 to the 4th hertz. It's a 10th of a millisecond time scale. You could ask, what's the neuron spike firing frequency down axons of neurons in your brain? And the spike firing frequency is around 100 hertz. Now, if you think about it in a quantum mechanical system, like a harmonic oscillator, there's a relationship between the frequency f of the oscillation and the temperature scale T star, which one could call a quantum to classical crossover temperature scale. So basically, you can define T star, which is Planck's constant times the frequency of the oscillation divided by the Boltzmann's constant. And for temperatures below T star, you're in the cold or quantum regime. And for temperatures above T star, the system is, if you will, hot and thermal, and presumably all quantum mechanical effects would wash out. So you can just plug in some numbers. If you put in a frequency of 10 to the 4th hertz and multiply by Planck's constant divided by Boltzmann's constant, you get T star is 10 to the minus 7 Kelvin. Really, really cold, way, way, way below body temperature. So one would conclude here that biological processes are thermal, not quantum. Basically, the body is too hot for quantum mechanical effects, right? So quantum mechanics should just wash out. But there's one caveat here, that this argument assumes thermal equilibrium. And if you think about the worldwide efforts to build quantum computers, the goal there is to isolate them, to have them not be in thermal equilibrium with their environment. They require, as I say, isolation. So let me just give you a 5-minute reminder of what a quantum computer will be once we've built them. So like in your regular classical computer, you have a bit, which takes on two values, 0 or 1. And in a quantum computer, that's replaced by the qubit, which could be the spin of an electron, a nuclear spin, or it could be any other two-state system. And the qubit is richer than the bit, since it can be in a linear combination of being up and down. And basically, a quantum computer that people are trying to build from the ground up consists of a number of qubits, which are then interacting with one another. And eventually, you would want many, many qubits interacting. And the key requirement for this quantum computer to be powerful is to have non-local quantum entanglement between the qubits. And this quantum entanglement must be created and controlled. So Einstein called this quantum entanglement spooky action at a distance. And the power of a quantum computer, loosely speaking, is that one can have multiple qubits which can be entangled non-locally. And this allows for non-local storage of information, non-local control, and non-local computation. And so that's all very good. But what's not so good is that a quantum computer will very easily entangle with the environment unless it's isolated from the environment. So isolation of qubits in a quantum computer is absolutely essential. It's paramount. And be that with superconducting qubits, solid-state qubits like NV centers in diamond, quantum dots, or topological qubits that one is being thought about. So isolation of qubits is paramount, And isolation of qubits is also very expensive. And because of that, the players in this game are mostly not the federal government, but the billionaire class, the Google founders. And Google's spending probably $100 million a year trying to build a superconducting quantum computer. And IBM is in the game, NEC, Hewlett-Packard. Microsoft is spending also in that scale of $100 million a year trying to build topological quantum computers. So it's always, I found this interesting that you have these software companies like Google and Microsoft where the only hardware they do is quantum hardware. But there are other software companies that could get into the game, and I'm still waiting for Apple to have its favorite platform for a quantum computer, or Facebook, or even Twitter. Now, before 2016, I didn't even know what Twitter was. Yeah, and now I wish I didn't. I don't know whether Rhode Island, where they went in the election, but I hope I'm not offending too many people here with that. OK, so we need isolation. So let's come back to the question of the quantum processing in the brain, the possibility of that. So quantum processing requires isolated qubits. OK, so what is isolated from the wet environment in biology? Are atoms isolated, molecules not really, proteins not isolated, electron spins are not really that isolated, and plus there aren't that many free electron spins. But the one thing that is isolated from the environment, particularly the liquid environment, are nuclear spins. OK, so protons and neutrons have nuclear spin a half, and some atomic nuclei, which are built from protons and neutrons, of course, have nuclear spin, which can take on values 0, 1/2, 1, 3/2, and so forth. As I say, these nuclear spins are very isolated from a wet environment. And you can see that by taking doing the following experiment, if you have an NMR rig at least. You can take salt, sodium chloride, you can dissolve it in water, the sodium and chloride ions separate, and you have sodium plus ion just floating around, solvated with water. And you can come in with nuclear magnetic resonance and basically measure the time it takes for the sodium nuclear spin to be disturbed by the environment. In NMR parlance, what you can measure are the T1 and T2 times, which in liquid state NMR actually, and in this context, are actually equal to each other. And what one finds is that the decoherence time, or the T1 or T2 times, is around 1/10 of a second. So 1/10 of a second is a long time on microscopic time scales. I mean, a water molecule bounces around in its cage at 10 to the minus 12 seconds. But on human time scales, 1/10 of a second is pretty short. But the nuclear spin decoherence time depends on which nucleus you're looking at. If one looks at lithium-7, and that's the common isotope of lithium, the nuclear spin decoherence time is a factor of 100 longer. It's 10 seconds. That's getting a bit longer. And what was remarkable when I saw this is that lithium-6, which is the rare isotope of lithium, which was purported to elevate the mood of the mother rats, the decoherence time is 5 minutes. My memory is not much longer than 5 minutes. And but 5 minutes, that's human timescales, macroscopic timescale. So nuclear spins can definitely be very isolated from the environment. And there's the rat reading the dictionary. Actually, we had, at one point, we had rats in our house. They were getting into our house, and my wife was pretty upset by it. And I wanted to start doing experiments on the rats. She said she would divorce me if I did that. My idea was to take some of the rats, feed them lithium-7, let them chill out in the living room and watch TV, and have the lithium-6 rats do the dishes. But it didn't work. OK, so quantum processing with nuclear spins in the brain. So the only way that we could have quantum processing in the brain is if it's with nuclear spins. So what I did at this stage, and this was soon after learning about these experiments on the rats with lithium, was to try to imagine that God came down and said, OK, evolution, well, I'm very clever, and I set evolution up so that quantum processing evolved in the brain. and reverse engineer it, figure out how that could possibly be the case. So I started listing a set of seemingly unrealizable conditions which would have to be satisfied in order for evolution to have had a path towards quantum processing. So we need a biological element with a very isolated nucleus spin to serve as a neural qubit. Now that's not lithium. Lithium's very rare in one's body unless one takes it therapeutically. So we need a common biological element with a very isolated nucleus spin. We needed mechanisms for quantum entangling the nucleus spins, mechanism for quantum processing with the qubits, method for quantum biochemical transduction. So the question was, can such conditions be fulfilled in biology? And I like to make this joke that as physicists, we know an enormous amount about next to nothing. But biologists know quite a bit about a lot, as do chemists. And I realized that I'd have to start trying to learn some biology and some biochemistry and some chemistry to see whether or not these conditions are actually fulfilled in the biological setting. And basically, I didn't dare work with graduate students or postdocs because I didn't want to destroy their career early on here. So my collaborators were Wikipedia and Google. And they're very reliable, particularly Wikipedia. That's Google, maybe not. OK, so let's just go down this list quickly. A common biological element with a very isolated nucleus spin to serve as a neural qubit. Neural qubit, question mark. OK, so what that actually requires is an atom with a nucleus spin that's spin 1/2. Nucleus spins that are larger than 1/2 are sensitive to electric field gradients, which are large in water, because the water molecule has an electric dipole moment. So nucleus spin 1/2 are only sensitive to magnetic fields. And one wants an atom that with nucleus spin 1/2 to be in a small molecule that can tumble in water to get to average out basically the effects of the magnetic field that are impinging on the nucleus. And one ultimately would like the other atoms in that molecule to have zero nucleus spin. So here I've listed the common biological elements, the biochemical elements, all single letters, CHNOPS, carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And the bioelectrical ions, sodium, potassium, calcium, chloride, and magnesium. And so the question is, which of these has nucleus spin 1/2? Well, the proton has nucleus spin 1/2, but I'm not so bold as to suggest that we've got quantum processing with the protons that are in water, which is what most of the body is made of. But of all these other common biological elements, there's only one which has, besides the proton, which is nucleus spin 1/2, and that's phosphorus. OK, now phosphorus has one isotope, phosphorus 31, nucleus spin 1/2. So the phosphorus is the only possible neural qubit. And if phosphorus 31 is not being used as quantum processing in the brain, then we don't have quantum processing in the brain. At least that's my belief. So this is very nice. There's only one atom we have to focus on. Where does phosphorus occur? Well, you probably remember, at least one of the few things I remember from biology was ATP, adenine triphosphate. Well, it turns out the phosphate ion is a phosphorus atom bonded to 4 oxygens in a tetrahedral arrangement. And so what you can do, you can take sodium phosphate, which is like sodium chloride, dissolve it in water. The phosphates are floating around in water, and you can measure the phosphorus nucleus spin decoherence time, the NMR T1 and T2 times. And one finds a time of around 1 second, which was disappointingly short. I was hoping it would be more like minutes or 5 minutes. And the reason it's not longer than this is because protons, which are there at physiological pH, tend to bind to the negatively charged oxygens in this phosphate ion. And the proton nucleus spin couples to the phosphorus nucleus spin. They entangle with one another, and that wouldn't be so bad. But what's bad is that these protons are hopping on and off the phosphate ion. So once the proton hops on, entangles with the phosphorus nucleus spin, and the proton hops back off again, the game's over. And that's what the relaxation mechanism is, to give this relaxation or decoherence time of only a second. But I wasn't about to give up. So what I asked is, what we need in biology is another positively charged ion, a cation, which can possibly outcompete the proton in binding to these negatively charged oxygens of the phosphate ion. So you can look at the other biological cations as sodium plus, potassium plus. They don't take part in chemistry at all in the biological context. There's magnesium 2 plus. Turns out that doesn't quite do it, but there's one remaining cation which is common, which is calcium. And it turns out that calcium 2 plus, I think will serve the function of outcompeting the proton in binding to the phosphates. And the hint for this comes from bone. Your bone is a mineral. It's crystal, and it's a crystal of calcium phosphate. And the story behind this so-called Posner molecule is quite interesting. In 1975, a man called Posner was looking at the X-ray crystal structure of bone, and he noticed that within the unit cell of bone were what he thought were structural units, and they were called after him Posner clusters, actually, consisting of 9 calcium ions and 6 phosphate ions in a particular, sort of almost spherical arrangement. This is a cluster which is a diameter of around 1 nanometer. Now, in 2010, there was some evidence from cryogenic transmission electron microscopy and dynamic light scattering that these Posner clusters actually can exist as free-floating molecules in simulated body fluids. And simulated body fluids are just water with the salt concentrations appropriate for your blood, essentially. And so if these Posner molecules are really stable in simulated body fluids and are stable in the body, they would be almost ideal for quantum storage and processing, because one would expect very long phosphorus-31 coherence times, because basically this molecule will tumble rapidly in liquid, and moreover, the oxygen, and I should have mentioned this, oxygen nuclei don't have any nuclear spin. The calcium nuclei don't have any nuclear spin. So the only nuclear spins in this Posner molecule are the 6 phosphorus nuclear spins. So where might these Posner molecules occur in your body? Well, it turns out the place that they might occur is in mitochondria. Now, mitochondria are organelles that you might have learned as the powerhouse of the cell. They're where ATP is produced. But mitochondria are really interesting. They're not just stationary organelles sitting there like a peanut, as they often are drawn. They're dynamical networks where they break into pieces, they fission and they fuse together, and they move up and down microtubules. Well, that's nice. But what's interesting from this context is that the mitochondria take in, and sequester, and buffer and release calcium from the cytoplasm in the cell. And in the mitochondria, the calcium is stored in calcium phosphate, a calcium phosphate sort of sludge. It's not actually, you don't have bone growing in the mitochondria, but it's a calcium phosphate sludge. So the conjecture is that Posner molecule, there we go. Posner molecule formation and melting inside mitochondria may be able to entangle non-locally networks via fission and fusion. So let me show you just some videos of mitochondrial dynamics. These are just from YouTube. If you look up mitochondrial dynamics, and let's see if I can get the video working here. And so what you will see, these are mitochondria, and the fluorescent dye. These two have just fused together, so the red and the green one became yellow. They're moving on microtubules. Here's a microtubule, and they're scooting up and down these mitochondria. And here's another sort of mitochondria moving on the microtubules. Mitochondria can also go from one cell to another through what are called nanotubes. Not carbon nanotubes, but organic nanotubes. Here's one other video. This may be a little hard to see unless you're right at the front, but basically these big blobs are the nuclei of a bunch of neurons, and if you can look closely, you see basically these microtubules, and these tiny little beads are mitochondria moving up and down. But I mean, I think that's almost impossible to see. But I'd encourage you to look up these mitochondrial dynamics videos on YouTube. So this is all fine and well. Let's say Posner molecules exist inside mitochondria. Mitochondria move around, they fission and fuse. But how could one do any quantum processing with these Posner molecules? Well, it turns out the way that quantum processing could occur is with what's called what I'm calling a pseudo spin. So the Posner molecule, based on quantum chemistry calculations, has a threefold rotational symmetry. And because of the threefold rotational symmetry, you can introduce a pseudo spin, tau, which takes on three values, 0 or plus or minus 1, which basically tells one how the nucleus spin state transforms under 120-degree rotation. And so this tau both characterizes the spin state and dictates the allowed rotational angular momentum states of the Posner molecules. So here L is the angular momentum, and L is tau plus 3 times an integer. So what's remarkable about this, though, is that if you look at the sectors with tau of plus or minus 1, it's not possible to construct a non-rotating wave function for a Posner molecule, because in order to get a non-rotating wave function, you have to get a wave function which is non-complex, which is real. It's like a standing wave. But you can't build a real wave function unless you have angular momentum L plus L and minus L. But when tau is plus or minus 1, you don't have L's coming in plus or minus pairs. So there's something very special then about stopping the rotation or not being able to stop the rotation of the pseudospin sectors where tau is plus or minus 1. So now the next part of the story is that based on, again, from quantum chemistry calculation, 2 Posner molecules are energetically, can chemically bond together. It lowers their energy, at least in vacuum, by about half an electron volt. And in work with Leo Radzahovsky, based on these pseudospin considerations, we conjectured that binding 2 Posner molecules implements a pseudospin projection where if the Posner molecules A and B chemically bond, then if they chemically bond together, there's a projection onto a pseudospin singlet, tau A plus tau B equals 0. So this gives one a mechanism for quantum entangling the pseudospins in the Posner molecules. In brief, so 2 Posner molecules are approaching one another, about to collide. They have some wave function psi, which describes their nuclear spin states. Let's say they chemically bond together for some time and then unbind. Well, that leads to a projection onto this pseudospin singlet. Alternatively, they can come together and try to chemically bind and fail to do so, and that projects onto the complement 1 minus this projector. In either case, 2 Posner molecules, when they've come together and either chemically bonded and then come apart or tried to chemically bond and been unable to do so, become pseudospin entangled. So this is a mechanism to induce entanglement between the pseudospin in Posner molecules. So the idea then is one has perhaps a network of pseudospin entangled Posner molecules, and moreover, the pseudospin entanglement can influence non-locally the pair binding. So if 2 Posner molecules A and A prime had bonded together and come apart, they would be entangled. And 2 other Posner molecules came together and bonded and came apart, B and B prime, they'd be entangled. So if some later time A and B come together and chemically bond, it turns out that then A prime and B prime are more likely to chemically bond than they would be otherwise, even though A prime and B prime have never been come close to one another before this binding event. So one gets this possibility for non-local non-local entanglement in chemical reaction. So, even if all of this is going on, it wouldn't be very useful unless there's some way to transduce the quantum, pseudospin and the quantum nuclear spin states into biochemical activity. And one can argue that the pair binding, which can be entangled, as I've just suggested, can lead to the dissolution of the Posner molecules into their calcium and phosphate constituents. And so this release of calcium can, in fact, modulate neural activity, creating possibly a quantum network that coexists with the neural network. So that's the, roughly speaking, the story that we've been developing. Now, are Posner molecules actually, do they actually exist? Are they really being used in quantum, in some quantum way? Well, I don't know, but Hollywood knows. And this is, unfortunately, the audio is not going to work for this, but there's a movie that maybe some of you have seen called Ant-Man and the Wasp. It's a terrible movie, I thought. But there's a little clip, and you can't really see this, but this is a famous actor whose name I forget. So, Paul Rudd. So he's saying, it's incredible you're linked to Janet. Oh, maybe. That's Fishbane. Yeah, we put quantum in front of everything. So even if you don't believe me, you believe Hollywood, right? They don't tell lies, right? So, okay, so what I've offered is a story at this stage. It's, if you want, it's like a partly formed picture puzzle. And what's needed are experiments to discern the precise shapes of the various pieces in this puzzle, and to see whether they actually exist as pieces, what shapes they are, and whether they start fitting together. And in the last year and a half, we've managed to secure some funding to try to look at some experiments, basically, to try to test some of these things that I've been talking about. And this is funding from the Heising-Simons Foundation. And there's a bunch of different experiments we've been doing, lithium isotope experiments on rats, enzymatic chemical reactions, Posner molecule, looking for Posner molecules, mitochondrial dynamics. Let me see, how much time do I have? I was told I should take 45 minutes.
That sounds good.
That sounds good, does it?
Does that work for you?
Can we ask you a question?
Sure, yeah.
Okay, so lithium-6 is 15% lighter than lithium-7.
Yeah.
And so then Stokes radius for diffusion is smaller. So then I would assume the lithium-6 ions diffuse much faster than, some percentage faster than lithium-7.
Although not much faster, because when they're in water, the lithium ion has very strong electric fields and it polarizes the water molecules. And there's a tetrahedral arrangement of 4 water molecules right around the lithium. And there's a second shell of water molecules with 12 water molecules. And so there's this big entity, which is actually pretty heavy. And whether it's lithium-6 or lithium-7 is not going to affect, very much at all the diffusion constant.
The biochemistry from the change of
The mass.
diffusion
Yeah.
constant, that everything will be split up a little bit, all the biochemistry will be changed a little bit. From that perspective, I mean, a more classical view, is it possible to explain?
Well, I don't know, but I mean, you have to contemplate that. And, but it's not clear what chemistry lithium takes part in at all. And maybe I will, okay, let me see. Since since I have a few more minutes, let me just skip through this. So don't look. Close your eyes. Oops, sorry. So I just want to list this because we've got some experiments going. So this, this is real science now. So at this point, it's been just storytelling, right? Of course, a lot of science is storytelling, but there's, it's really an experimental endeavor, of course. And so is there really a lithium isotope effect on rat behavior, independent of what the explanation of it is? And so we have some new data on a ketamine rat model of mania. Do nuclear spins modulate chemical reaction rates? That's really a crucial piece. There's one particular chemical reaction we've been looking at, not because it's so relevant to the quantum brain story, but just that it's a molecule with nice threefold rotational symmetry where there's an enzymatic chemical reaction that we can look at and see if there's a carbon-13 isotope effect. And we've got some preliminary results which are looking very interesting there. Can we detect Posner molecules in vitro? I mean, do they actually exist outside of Hollywood? Are Posner molecules inside mitochondria? So these are the types of questions that we're seeking to answer. And I won't go into any of the details here, but here's an experiment I was involved with of lithium isotope effects on rat behavior, where basically the rat model for mania is to inject rats with ketamine. So I don't know if you've heard of ketamine. Maybe it's an anesthetic when it's at high concentrations. It's a recreational drug called Special K when it's at moderate dosages. It's a treatment for depression when it's at lower dosages. So it's an amazing drug. With rats, when you inject them with ketamine, they run around a lot. It makes them manic. But if they're on lithium, the lithium counteracts the ketamine-induced mania. And what we found was that lithium-6, the rare isotope of lithium, is even more effective at counteracting the ketamine-induced mania. So these blue curves here are lithium-6, and this is the distance travel as a function of time for these rats running around in an enclosure. Here's dynamic light scattering experimental data where well, I'll just show you. I mean, look, there's basically by looking at the autocorrelation function of the light scattering, one can detect or measure the diffusion constant for particles or molecules floating around in the solution. And using Stokes-Einstein relation, one can go from the diffusion constant to a size scale or a diameter scale of the molecule. And in dynamic light scattering, which was done with beautiful work by Josh Straub, who's a graduate student in the physics department at UCSB, he sees a very nice peak here at about 1 nanometer. So this is consistent with these Posner molecules. We now want to start doing NMR to look at the phosphorus nuclear spins in these Posner molecules. We want to start doing what's called cryogenic electron microscopy to actually look directly for these Posner molecules. And I think what I will do now is go back and just finish up with a discussion of the future of quantum computing. So when will we have a multi-purpose quantum computer? Well, what's the path towards trying to build a quantum computer? It's pretty short, right? It started really in 1982, where Feynman suggested that quantum computers could do interesting things. They could simulate quantum systems, is basically what he was suggesting. Peter Shor in 1994 developed this remarkable algorithm showing that if you had a quantum computer, it could prime factorize numbers much more quickly than any conceivable classical computer. And so on. And so basically, it's really only the last 5 or maybe 10 years that there's been a huge effort to start actually building quantum computers. So multi-purpose quantum computers, how long will this be? I mean, it depends on how optimistic or pessimistic you are about the state of the world today. So I'm pretty pessimistic. So I think it's going to be a long time, 50 years. That's well after I'm going to be dead. But maybe it's going to be 10 years. Maybe it's 20. Maybe it'll be never. But let's just say it's 50 years. So the path towards a quantum computer is going to take some 50 years or so. But I want to take a longer view towards the path of a quantum computer, starting 3.5 billion years ago. Single-cellular organisms developed, multicellular organisms developed, plants developed, invertebrates, vertebrates, mammals, humans. Google human engineers eventually evolved after 3.5 million years. Now, Google engineers are very good classical robots. And they're building a quantum computer. And maybe in 50 years, if they have one, whoa, evolution would have figured out a path to evolve a quantum computer by first evolving clever Google robots. So how many years will it take for evolution to design or build a quantum computer? So there's two choices here. 3.5 billion years plus 50 years, or another possibility is just 3.5 billion years. And you take your pick. I don't know which it is, but we'd like to answer that question. And so the main thing I want to leave you with is that it's a very wild question that we've been pursuing. Is there quantum processing in the brain? The brain is extremely complicated. Biology is extremely complicated. Quantum mechanics is extremely complicated. But it's very constrained in our reverse engineering. And the question, I think, is in principle answerable with experiments. And we're determined to try to do as many experiments as we can to try to answer the question. And maybe, by 50 years from now, we will know that indeed it was just 3.5 billion years that it took to evolve quantum computers, and that we can be very proud of the quantum computers that's sitting between our ears. So anyway, thanks so much for your attention.
We have time for some questions.
Yes, I'm curious about if there are, are there any implications for let's say, memory or function, with the fact that there are these three-level spin states, or opposed to two or whatever?
Yeah, I think whether it's a Q-trit, which would be like a three-level system, or two, is not so important. But I get asked, what good would it be if we did have quantum processing in the brain? So what? How would that be helpful? Well, it would give one another parallel channel of information processing and possibly information storage. But it's also possible that Roger Penrose maybe wasn't so far from the truth, and that in order to have sentience and emergent consciousness, one would actually have to have a quantum computer. And if that were true, it would be very beneficial, of course, to be sentient. I mean, for us, at least most of the time, it feels that way. But I think whether it's three states or two is not so important.
I guess kind of going off of that a little bit in terms of like the evolution or our evolutionary history that led us to this point, what's the implications, or in your opinion, what are the implications about other species, like our close relatives, in terms of quantum computing and their potential to have those capabilities?
Yeah, that's a great question. And so, of course we don't know, but let me speculate a little bit on that. So, I talked about, so let's assume some of the story is correct. So mitochondria are important. Well, single cellular organisms like a bacteria don't have mitochondria. Okay, so if mitochondria are important, then single cellular organism wouldn't be able to have quantum processing. Now, the calcium phosphate is part of bone, so the vertebrates, the animals with bones, have very refined calcium and phosphate modulations. Basically, the concentrations of calcium phosphate are very important. So, so maybe it's the vertebrates, which is where the quantum processing started. Well, what about mammals? Well, it turns out the main ingredient in mammalian milk is calcium phosphate. You've heard that calcium is good for the brain, and breastfeeding, is good for the child, the cognitive development. I mean, that's and as I say, mammalian milk is a way to deliver essentially liquid bone, to deliver calcium phosphate. So maybe maybe there's different levels of quantum processing, as you go through the evolution, history of evolution. Just like you can have a quantum computer with a smaller number of qubits, that or the speed of the operation of the qubits is slower, and then you can have a more refined and better quantum computer. So, but very good question. Thank you.
Yes, Brenda.
So in order to have information processing, you need to read, write, and also do those things in a non-stochastic way.
Yeah.
So far I've heard lots of stochastic ways. How are we doing the reading, the writing in the non-stochastic way?
Okay, the non-stochasticity, it turns out that within the the rules that are basically described about these Posner molecules chemically bonding together and coming apart. If you take a whole collection of Posner molecules and you let them start evolving stochastically so that they come together pairwise, sometimes they chemically bond, come apart, sometimes they don't chemically bond, that at long times, even if you start in a like what's called a quantum mixed state, at long times the quantum state purifies, and it can go into a cat state where there's a linear combination of all of them having pseudospin up plus all of them pseudospin down, or a state with all of them having just pseudospin up or just pseudospin down. So there's actually, by having projective measurements going on during these pair binding of Posner molecules, the projective measurements can actually what's called purify the quantum state and lead to pure states, and essentially lower the entropy. So stochasticity can work in your favor in this case. Now, well, and the I mean the reading and writing, I mean, are basically writing into the entanglement and how quickly these Posner molecules are going to be chemically bonded together or not depends on the pH. And the pH inside a mitochondria depends on how much energy is being burned. It turns out the pH gets modulated during ATP production. And so there's a very, those are, actually tied together. And the readout is under some conditions, the calcium and phosphate can dissolve, and the calcium can be released from the mitochondria. And calcium is one of the most important ions in neuroscience, and it triggers biochemical activity. So there are bits of the pieces of the puzzle. One can tell a story. Now, a lot of this is based on things that need to be checked experimentally. First of all, do Posner molecules exist? Do they chemically bond together? When they chemically bond together, is it equivalent to making a projective measurement onto the pseudospin singlet? And so forth. Are Posner molecules in mitochondria? Can you get situations where the mitochondria can become entangled? And so there's many pieces to the puzzle that need to be looked at. But yeah.
If I could, just maybe another way of looking at your evolutionary predictions here. Through the snapshot of your talk today, it's got to do with calcium and calcium concentrations. There's kind of a numbers question here, in that you have talked about the Posner molecules and in a very elegant way proposed this quantum entanglement, which then would lead to some biochemical pathway, a transduction of this proposal, into some functional outcome.
Yes.
That affects the brain or some aspects.
Exactly.
Of the brain.
Yeah.
Now, as you know, there are any number of much more classical sort of views that have to do with the concentration that we know of not only calcium but especially sodium, potassium, and so on, which are really the apparatus which make brain dynamics, potential, and so on.
Yeah.
So I'm wondering, what might be even the de facto concentrations of calcium out of the normal calcium that might participate in that, and in that sense, if you wait another 10 million years, might that, degree of calcium that's really imperative to your argument somehow manage to make its way even into a more present modulation of brain?
Yeah. Okay. Well, maybe let me try the following with calcium. I mean, so like if an electrical signal goes down the axon of a neuron, an action potential, when it reaches the synapse, calcium channels open. The calcium concentration in the cell, in the cytoplasm inside the cell, is very, very low. It's much higher outside the cell. So when these calcium channels open, calcium flies into the cell, and the calcium near the synapse then, basically complexes on these vesicles which store neurotransmitters, and the neurotransmitters are released into the synaptic cleft, and then they can cross the synaptic cleft and trigger neuroreceptors on the postsynaptic neuron. But after that's happened, there's a lot of extra calcium which is floating around inside the cytoplasm, which has to be cleaned out. And there are calcium pumps which pump the calcium out of the cell, but also the calcium gets sequestered into the mitochondria. And the more you have the calcium that comes in, the more calcium gets sequestered. But it doesn't continue to get sequestered indefinitely. It gets released at some time when it's needed. And calcium, when it's released from the mitochondria, can trigger all sorts of biochemical activity, including perhaps neurotransmitter release, or it can maybe enhance the neurotransmitter release. So the fact that calcium came into the story it was fortuitous in a way, and it came in because it can bind nicely to the phosphates and protect the phosphorus nuclear spins. But then it's also great for quantum to biochemical transduction as well. I don't know if that.
So I have kind of a curiosity question. So you mentioned that these quantum computers need to be isolated from their environments.
Yeah.
So I guess in what way do they need to be isolated, and do you think that it'd be possible to one day make a laptop or a mobile device a quantum computer, or will it never achieve that level of isolation?
Well, they need to be so. So most of the efforts in the laboratory to build quantum computers involve thermal isolation, so cooling the system down to very low temperatures, like superconducting qubits or ion traps. I mean, they cool down to very low temperatures. There have been efforts to do what's called liquid state NMR quantum computing, which is just in a liquid at room temperature with using NMR to try to manipulate the nuclear spins on molecules. And so that's, so I mean, if there is actually quantum processing going on in our brains, evidently it can happen at body temperature. I mean, and then one might imagine being able to build an artificial biological quantum computer, which you can hold in in your iPhone or something, iPhone star. I don't know what it would be.
So one of the things that the warning signals was going off in my head when I was listening to the talk was that large molecules, when they rotate in liquids, aren't quantum mechanical by and large, in the sense that angular momentum isn't a good quantum.
No, I know.
You don't really see all the stuff that shows up in the beginning physics courses about rigid motors and stuff.
Yeah.
With large molecules.
Yeah.
So wouldn't it be a better strategy in terms of thinking about how one might get quantum computation to stay as much as you can with the nuclear spins and don't try to work with, assume that you're ever going to get nuclear motion being quantum mechanical in liquids?
Well, okay, so it turns out, I mean, angular momentum is not a good quantum number, but this pseudo spin is. The angular momentum mod 3, if the molecule has threefold rotation symmetry, is a good quantum number. And so, so it does seem surprising that you could have this, the chemical reaction rate, let's say, depending on the pseudo spin value. I mean, I think the alarm bells that were going off would maybe suggest that that's not possible. But we're going to test that in these threefold rotationally invariant molecules, trimethylamine oxide, by looking at the carbon-13 isotope effect. And so, this is something we can really put to test. And but I think we need to use this. In order to get the nuclear spins to, which are quantum mechanical, to biochemically transduce, we have to go through something like the rotations of molecules. I don't see any other way to do that. And so, maybe that's not possible ultimately, in which case then that's maybe the end of the story. But it's testable. We can look into directly at least on the nuclear spin or the pseudo spin effects on chemical reaction rates by looking at isotope effects. Which is what we were doing.
I'm going to do one more and then.
What sort of brain functions would require us having quantum processing capabilities?
What brain functions? Well, if it's sentience and awareness, maybe it's the brain function that's needed for you to ask such a good question. I don't know if I can do better than that.
Well, we know that quantum computers would be good for factoring numbers.
Yeah.
That we don't seem to be very good at doing.
No, exactly.
It's just that I see that what we currently know a quantum computer could do would seem to have little use for an organism that evolves in natural state.
Well, I mean, it's true that we only have basically two algorithms for quantum computing, Shor algorithm and Grover's algorithm. But, you know, I think the lack of algorithms that we have for quantum computers is because it's probably a limit of our imagination. I mean, you know, I think if we actually had a big quantum computer that we eventually will have, it's going to be able to do things that we haven't even anticipated. That's, I hope, and I expect. Otherwise, it'd be a bit of a shame, because if it's just prime factorization, it's that's a lot of money to be spent. But
Thank you very much.
okay.